and welcome to today's webinar. My name is Donna and I work as a help desk bioinformatician in the EGA team here at EMBL-EBI. My colleague Giselle will be available via the Zoom Q&A box for any questions you may have. So in today's webinar, we will cover some general information about the EGA, the data at EGA, how to go about accessing the data that interests you, duo codes at EGA, and finally, how to download the data from EGA, which we are going to demonstrate using our EGA test dataset. So just a small history about EGA. The EGA was established as part of EMBL-EBI back in 2008, and in 2012, we started to work with CRG in Barcelona. So as you know, EMBL-EBI has many archiving services. So what makes EGA different? So we have ENA, that is the European Nucleotide Archive. So the ENA records raw sequencing data, sequence assembly information, and functional annotation. Then we have the Array Express, which is a database of functional genomics. We have the EVA for storing variation data, and we have the Biosamples, which is a database describing biological samples. So all these resources are open and public archives. EGA, on the other hand, is a controlled data access archive, providing distribution of genetic and phenotypic data. So what is controlled access data? So participants in medical or genetic research projects have typically provided consent for their data to be used in research, but not for open public distribution. And these data require a secure archiving, processing, and distribution service that respects the original informed consent agreements. So without consent, the data cannot be generated or shared. And thus, the EGA was created as a service to make sure that all such data can be made available for the researchers who have been granted access to the data in a secure and controlled way. So in order to encourage the participants to share their data, it is stated within the consent form that data sharing will only take place under controlled or managed access condition. So it's because of all the above that the data requester would have to make a formal access request to the governing data access committee or the DAC for accessing a dataset. So now we know why EGA was established. Let's take a little look inside of EGA. So the screenshot that you're seeing now is taken from our homepage. As you can see, cancer studies comprise around 40% of all the studies at EGA, but we hold a very wide and diverse range of other studies ranging from arthritis to malaria. Now here is a live demo demonstrating how to search and request for data at EGA. So this is our website, and let's say I'm a researcher and I am interested in downloading any data that is related to COVID. So all I need to do is type the keyword COVID into the EGA search bar that you're seeing here. So as you can see, currently there are 12 studies in EGA that has data related to COVID. So you are seeing the study title right now, and then you can choose the study title of your interest. So let's choose this study title. Now we are being directed to the study page. Here you have the study description. So in the study description, you get information such as how the study was done, what experiment was done, what files were generated, what protocol was done, etc. You can read in the study description. And then you can see the data set that is associated to the study. So in EGA, studies are referenced by the EGAS accession and data sets are referenced by EGAD accession. At the end, you can also see the publication that is associated with this study. Now let's click on the dataset link. So the dataset link will bring us to the dataset page. Here we can see the dataset title, the dataset description, as well as what technology was performed or used to get this dataset, how many samples are there in dataset. All those information we can get from the dataset. And most importantly, to get access to this dataset, we are being shown the data access committee for this dataset. So if I'm interested to gain access to this dataset, all I need to do is make a formal email to the DAC with my request, and then they would be assessing my application and asking me to fill a data access agreement, which I need to fill and send back to them. And once those processes are done, the DAC would email us at EGA, and then we would be creating an EGA account for you so that you will be able to download this data set of interest. You can actually search for any data set, any disease of your interest. For example, brain cancer. As you can see, currently there are about 42 studies at EGA that has data related to brain cancer. So that's about searching and requesting for data at EGA. So now the second part of the webinar is regarding data use ontology. So data use ontology, or in short DUO, is an ontology which represents data use conditions. So DUO codes allow EGA to tag dataset with restrictions about their usage. Now let's see how DUO codes help. So let's say we have a research paper of interest, and we see in the paper that the sequence file have been deposited at the European Genome-phenome Archive under the accession codes, and then 2 data accession codes are given. Now let me just go to the link of the dataset in EGA. Yeah, so this is the dataset, and if you scroll down here, you will be able to see the DUO codes that are tagged to our dataset of interest. So further information on each DUO code can be obtained by clicking on the respective code links. As you can see, this dataset is associated with 5 DUO codes such as institution-specific restriction, user-specific restriction, publication required, research use only, general use in clinical care, all these things. And if you want to know what each DUO code represents, just click on the DUO code. So DUO code 26 basically means that the use is limited to the use by the approved users. So in simple words, that means that if a user is given an access to a data set, that credentials should not be shared with anybody else. That credentials should remain private and should be only used by the user who has been approved by the data access committee. And thus, the DUO code will help the data requesters to identify how the data may be used and if they would likely get access. The DUO code also helps to mitigate unnecessary application to the data access committee. So suppose I'm a researcher and I want to perform general data analysis in bioinformatics. So I search in EGA and I find my data set of interest at EGA. So this is the data set that I found in EGA, and I found this data set interesting. So let's just go to the data set link at EGA. So this is the dataset link at EGA, and as you can see, it's deep sequencing for circulating tumor DNA determination. And then I go to the Duo section and I see that this data set is associated with Duo code 7. So I can see that the label that is given to this Duo code 7 is disease-specific research. So let's investigate this Duo code further. So the definition for the Duo code is given is that the use is allowed provided it is related to the specified disease. Now we need to find out which is the specified disease which the submitters are referring to. For this, the submitters have given us the Mondo identifier. So just copy paste the Mondo identifier and then go to the Mondo service page, which is also another service provided by EBI. Paste this Mondo code, and then we get the result as colorectal cancer. So I should be applying my access to this data access committee only if my research is specifically regarding colorectal cancer. So that is why this Duo codes actually helps to mitigate unnecessary application to the data access committees. Now, the third usage of Duo is that scientists can search on the Duo codes aiding data discoverability. So if you go to the EGA site, in the search bar, you can also search with the Duo code. So if you type Duo, all the Duo codes will appear. And let's search for Duo code 18. So currently there are 73 data sets in EGA with the Duo code 18. And you can see all this data set title here. So let's take this data set, that's Internation RNA-seq data set. And you can see that this data set is associated with many Duo codes. And Duo code 18 means that the usage of this data set should not be for profit, and it should be for non-commercial use only. So like this, you can find your Duo codes of interest and you can search on EGA and you'll find the data sets that's associated with the Duo code. So the final part is like how to download the data at EGA. So once you have gained the permission to the dataset of your interest from the appropriate data access committee, you can now proceed to download the data. And this is done via the EGA download client or the EGA 3. So the EGA 3 download client is simply called the Python download client. So from the name itself, it's a Python-based tool for viewing and downloading files from authorized EGA dataset. So the Python client is compatible with any operating system, and you should be having Python 3.6 or higher versions installed. And the Python client only supports command line use. And the three major requirements would be connection to the internet and sufficient space on the destination drive, as some data sets can be very huge, like it could be 10 terabyte or some data set could be 50 terabytes. So it's important that you have sufficient space on your destination drive. And of course, you would need an active EGA account. In case you don't have an EGA account, you could use the EGA test account that we have prepared using data from 1000 Genomes project. So talking about the login into the Python client, it can be done in two ways. The first one is via a credentials file, and the second one is through an interactive command line session. So first we'll see how to log in using a credentials file. So defining your credentials file. So as you can see, this is the template for the credentials file, and you can get this template from our GitHub page. So we have provided the link here. So this is the template for your credentials file. All you need to do is just copy paste this template and open any text editor of your choice and paste this template. So the username would be your EGA registered email address and the password would be a respective password. So here we are using the credentials for the EGA test account. So these are the username and password for your EGA test account. Now next, we should proceed to save this file in .json format. So I'm just saving this file in my desktop. So always make sure to save this file in .json format because that is how the Python client would read the file. So that is saved in my desktop. So next is about the Python client installation. So the Python client installation is very simple. You can achieve that by typing pip3 install pyga3. But we kindly ask you not to try installing the Python client now, as depending on your Python version installed or your operating system, or even the installation of pip3 in your operating system, you could face some difficulties and thus you may lose track with the demo of the Python client. So we would kindly request if you could try installing it after the webinar. And if you have any questions or any problem with installation, you can reach the EGA help desk team and we will be very responsive to your issues. So here all you need to do is open your terminal and install the client. So since I've already installed it, I'm getting reg requirement already satisfied. So next is about using the download client. So if you are new to the EGA download client, it will be very beneficial to know the different identifiers that is there in EGA. So the two major identifiers are EGAD and EGAF. So EGAD stands for the EGA dataset accession ID. That is the unique accession ID that we have given for each dataset. And EGAF stands for the unique accession ID for all the files in the dataset. And the Python client also has different arguments or different parameters. And we'll look at each of them. So -cf stands for credentials file. So just a while ago, we saw how to define your credentials file. So this credential file is called using the -cf argument. So it's the JSON file containing the credentials. So if you want to list the data sets which your account has access to, the datasets argument is used. And if you want to list all the files in a specific dataset, the files argument is used. And if you need to fetch a dataset or a file, that is, if you want to download a dataset or a file, the fetch argument is used. And the next is the -c parameter or the connections parameter. So the connections parameter helps us to download using specified number of connections. For example, if I want to download a dataset using 20 connections, I should be using -c20. If I want to download using 30, it would be -c30. And finally, the -d, that is the debugging. This would give us the extra debugging messages that can be helped for the EGA team to debug any issues that you might be having. So we'll just quickly go through a live demo of the download. So let's say I'm interested to see what are the data sets that is there in EGA test account. So all I need to do is to call the Python client And now we have to, so let me just cd into my desktop because that's where I kept my credentials file. So we call the Python client, and then now we have to reference the credentials file. So as we saw before, the credentials file is called using the -cf parameter, and then now we'll call the credentials file. And I'm interested to see what all data sets are there in the EGA test account. So just call PyGA3, and then we reference the credentials file, and then we give dataset. So this would start the session. As you can see here, you can see what is the client version that you're having. And this is our latest client version. So always make sure that you are on the latest client version, or else you will have problems in the download. And this shows the Python version that is installed in my operating system. As you can see, it's 3.9, and the OS version, that is what is my OS. And finally, it has given me the message that authentication was success. And now it has given me the data sets that is under the test account. As I mentioned before, these data sets have been derived from the 1000 Genomes project. So these are publicly available for anybody to download. Now I'm interested to see what all files are there in this data set. So the same command, that is PyGA3, we are calling the Python client, and then we are referencing the credentials file, and then here files followed by the dataset ID. So the same process again starts, and the authentication is done. Yeah, now it is showing me what all files are there in the test data set, this one. So it is showing me all the files, the file ID. These are unique accession IDs. And bytes means what is the size. So this is the size of this file. And checksum is basically used for checking the file integrity once we have downloaded the file. And then we have the file name. So as you can see, this data set contains scribe files, it contains BCF files, VCF files, and BAMP files, and its index files, it is the BY files, and a lot of CRAM and their CRY files. So all these are there in the this test data set. Now let's try downloading a file. So I'm interested to download this file. So it will be PyGA3-CF credentials, and we use the fetch command. So as you can see, the Python client itself is telling me what is the space in my operating system, and what is this, how much space I've used, and how much free space I have. So this is very useful when you are like trying to download a file and it's very huge and you might run out of space. So it's a very useful feature of the Python client. Yeah, so now the download has finished, and the download has been saved to my desktop folder. And then this is the size of the file, and the MD5 value is also being displayed. So I can cross-check with the MD5 value here to see if the download has been successful. So that's how you download a file. Now let's see how to download a dataset. So the same process follows. So instead of the file ID, we just provide the dataset ID. So let me just get the dataset ID for you. So here, all the files in this dataset would be now downloaded for you. So the download has started. One file is already downloaded and the second file is being downloaded. So it will go on till the last file in this data set is downloaded. I'll just stop this process. And if you are ever trying to download this test data set, please be mindful that it is almost a terabyte. At the end, you can see how much the whole data set is, and it's almost 1 terabyte. So please be mindful when you try to download this. And also, if you are interested, you can also download this whole data set using the connections. So if I put -C20, that means this download will take place using 20 connections. And if I put -C30, the download will take place using 30 connections. Yeah, so if the file already exists, the Python client doesn't download it again, but it starts with the next download. So I'll just quickly end the session. So all the commands that I've used now is put in our presentation, which we'll be sharing with you. So to display authorized dataset, we are just calling the credentials file and then dataset. To display files, it will be files followed by the dataset ID. And to download a single file and to download a whole dataset. And you can download a file or dataset using N connections. Now next is download using interactive mode. So in case you are not interested or you find using a credentials file very time consuming, you could also use the interactive mode. So to use the interactive mode, it would be PyGA3 datasets. You don't have to specify the credentials file. So just press enter. And then it will ask for the username and password to be typed in the interactive command line. So I'll just input the username for the test account, which is the EGA test data at ebi.ac.uk and the password. So the authentication is success, and it has displayed for me the data sets that are under the test account. So we at EGA have also hard-coded this test data set using -t parameter. So how to use that would be just type PyGA3 -t datasets. So this username and the password is already hard-coded into -c. So if you are using the test dataset, you can just mention -t and Python client will understand that you are referring to the test account. And similar wise, if you want to, that's it, I think. So that's the download using interactive mode. You just have to mention PyGA3 datasets and then PyGA3 files and followed by the dataset ID, PyGA3 fetch followed by the file ID, or if you want to download the dataset, it would be PyGA3 fetch followed by the dataset ID. So in case of any download failures or errors, you can use the -d parameter. So the -d basically means debugging. So it will give us extra debugging messages. For example, let me just put -t and show you. Just so it will just print us for some extra debugging messages. That would be if you have any connection issues or anything, if you could forward us the output that you get with -t, that will clearly help us or the developers or the help desk support to understand what is the issue that you're having. And if any the support that we could be providing is really helpful if you could provide us the output with the -t. And also if you have any queries, and if you try using the test user account, you could reach out as at helpdesk@egaarchive.org.